Hi, my name is Dedi Meiri. I'm a professor in the Technion, the Technology Institute of Israel, and I'm leading a laboratory for cancer and cannabinoid research. And actually, my lab is working around the medical cannabis properties, and you're listening to Professionally Cannabis.
Where do they go, the smoke rings I blow each night? Oh,
Shalom listeners and shalom Oscar, and welcome to Professionally Cannabis.
Shalom, Jonathan. Welcome to the new year.
Well, it's really exciting to be here. A new year, a new season, and many new guests. The reason for the peaceful welcoming with Shalom is that our guest for today, I'm really honoured to say we were joined by Dr. Dedi Meiri. Oscar, tell me a little bit about him.
Well, this has actually been one of my fanboy guest extravaganza wants since we began. But I won't jump the gun on too much of the interview, but Dr. Meiri's work, especially with regards to certain cancers, really has just pushed the envelope forward on what a cannabis medicine can do. What he's done is he's drilled down into the fact that cannabis is not a single drug, but a mixed bag of 500 active components, and he's proven in a variety of different ways that for the right cancer with the right cannabinoids and or other compounds, you can see really effective results in vitro and in mice. And so taking this forward, bringing it to clinical trials and taking it to humans, it's really going to upend the medical establishment. I was super happy that we managed to get this episode.
And as a heads up, during the interview, there are moments when you can hear his team in the background working hard. Hopefully, we can forgive it for this edition, seeing as they are literally trying to cure cancer.
We had so much good stuff in this one, we split it into two. So enjoy part one.
It gives me great pleasure to welcome Dr. Dedi Meiri to Professionally Cannabis, heading the Laboratory of Cancer Biology and Cannabinoid Research at the Technion University, Israel's Institute of Technology in Haifa. Much of Dedi's work is focused on the effects of cannabinoids, the chemical compounds found in different strains of cannabis, and the movement and growth of cancer cells in the body. He's been referred to in the past as one of the greatest minds in cannabis, something that I wholeheartedly agree with. Dedi, welcome to the podcast today.
Hi, thank you very much for these warm words, and I'm pleasure to be here.
Thank you for joining us. So, I don't know if you've heard our podcast before, but we like to start with a common question to all our guests, which is, what led you to the plant?
So first of all, I'm a son of an agriculture, and we had a big farm, and I was agriculture all my life. I have a PhD in plant molecular genetics, so my background is from plant. And when I established my lab for cancer research, after I did my postdoc in this area, I wanted to combine plants with my research in cancer. I thought I will have a kind of advantage as a person that all his life spoke with plants. So cannabis was just there. It was almost 5 years ago. It was still a desert. Nobody did the research on that. But there was few publications by Manuel Guzman and Christine Sanchez from Spain that showed that cannabis can attack cancer cells or inhibit their proliferation and migration. And this was just a fit for me. It was biochemistry, it was a plant, it was cancer. So it was a match, and then I started to look deeper and deeper.
So is it fair to say that you didn't have significant exposure or knowledge of cannabis before you came to it from this oncological side?
No, actually, almost not at all. For 7 years, I was the head of the Botanic Gardens in Tel Aviv University, and I worked a lot with medical plants. There is a big greenhouse for medicinal plants. But as you can assume, cannabis is not part of them. So actually, I was quite surprised to learn that it's one of the most significant, most known medical plant. And actually, until I established my lab in Technion, I didn't even know about it. Of course, I know what it's cannabis, but on the recreational side, but I didn't aware at all about its medical effects. So I was quite surprised to learn that.
And if possible, I'd like to start us somewhat before the time of which you started your lab in Haifa. You know, I understand that you spent 4 years in Canada at the Ontario Cancer Institute. Can you tell us a little bit more about the time you spent in Canada and how perhaps that shaped and provided the platform for the amazing work you've been doing later on in the Technion?
So I worked in the hospital and I worked in the research. And for 4 years, I was doing study mainly on pancreatitis and breast cancer, trying to understand how cells started to migrate and to invade. So first, cancer is a given name to hundreds of different diseases. We put them in a box. We call it the 6 hallmarks. Every time that we see 1, 2, 3, 5, 6, we will call it a cancer. And normal cells in our body don't have ability to move. We have cells like macrophages that have ability to move, but let's say skin cells, pancreatic cell, breast cells, they are cells that don't have ability to move at all. But when they become cancerous, it started to develop the ability to move, to migrate, and to invade to different areas, and we call it metastasis. So my focus in my research was how cells that don't have the ability to move started to get this ability and started to grow, and there is big changes in compounds called cytoskeleton of the cells that change. And actually, this is what leads me also to the cannabis, because I saw a publication by a Japanese group that showing that putting cannabis on breast cancer blocked the ability to migrate. And this was, I said, wow, okay, this is a fit. It's a plant, it's biochemistry, it's blocking the ability to migrate, which was my professionality. And that's the connection here.
Okay, and so that's the start of cannabinoids and cancer for you. And can you give us a brief history of where you've come to over this past 5 years? What's the latest and greatest in cannabinoids against cancer? Because I think for many of our listeners, they'll be aware that there can be anti-tumor properties or anti-metastasis or even anti-angioplasty, if I've pronounced that correctly. Where's the latest?
Yeah, so when I saw this publication that cannabis blocked the migration, I was very excited. But then, as I said, I started to read papers, and I saw papers by these groups from Spain, Manuel Guzman and then Christian Sanchez, that showing that putting cannabis on cancer cells can cause apoptosis, actually can bring the cells to commit suicide. As I said, cancer, it's a given name to hundreds of different diseases, and we put them in a box. We said 1, 2, 3, 4, 5, 6, it's a cancer. The first thing that happened, these cells losing the ability to commit suicide. So every cell in our body have machinery called apoptosis, and when it's realized that there is kind of a problem or mistake, there is a checkpoint when cells proliferate, they will commit suicide. Think about it. Cell, it's the basic unit of life. So we have life that are one cells, like bacteria or yeast. This is one cell, which is a creature. Also in our body, every cell, it's like a unit. If you're taking one cell from our body and put it in a plant, it can grow and proliferate and move and can do everything alone. But in our body, they must communicate with each other. They're working as organs, and they need to communicate with each other. If the cells realize that there is a mistake, there is a problem, he will commit suicide in order not to hurt the other cells around him. And this is in every minute in our body. There are thousands of cells that are committing suicide. But when there is a problem and the cells have a problem in this machinery, in this apoptosis machinery, or he knows how to avoid this, this is the beginning of cancer. So Manuel Guzman and Christian Sanchez show that if you put cannabis on these cells, it's bring them back the ability to commit suicide. So this was a problem for me. The Japanese show that it's blocking migration, but this showing that they are killing the cells. So the question was, okay, does it kill the cells? Does it block their move? Which type of cancer? Breast cancer, liver cancer, pancreatic cancer, prostate cancer? Is it the same? And for the other hand, what the hell is cannabis? Cannabis is a given name to hundreds of different species or different plants. Today in my lab, I have more than 900 different types of cannabis. So which one to start? So we started doing kind of matrix. We said, okay, let's take 8 different types of cancer, 8 different types of cannabis, and let's see how it's affecting. And we were very surprised to see that when we use extract number 1 from plant number 1, it's killed the breast cancer, but extract number 2 from plant number 2 didn't affect the breast cancer at all. And when we look on prostate cancer, the one that killed the breast cancer didn't affect the prostate, but the one that didn't kill the prostate, we saw it was a mess. And we were very excited because, first of all, we saw a very severe reaction of the cancer cells, but maybe I think the amount that we had to put on the cells in order to kill them was very surprising. In the end, every material will kill cells in the plate. If you take sugar, if you took salt and put on these cells, the cells will die. But there is something we call IC50, how much we need to put in order to kill these cells. And I was surprised to see that the amount was minor amount. Actually, the same amount that you need to put the most severe chemotherapy and sometimes even less, which meaning that it's extremely, extremely potent and extremely, extremely active. So now we were excited and want to see which compound, what in the cannabis doing that effect. I learned about the cannabinoids. I knew that there is THC and CBD. So I said, okay, let's send it to a laboratory to check it. I'm talking about 4 years ago. How you send cannabis? You don't send cannabis in the mail. Like, which laboratory have the ability or license to do this examination? 4 years ago? 0. My license to do cannabis research in Israel, it's R for research, 0001. So I was in the desert. Nobody can help me. I have in Israel Rafi Meshulam that probably most of the audience know the name. He's the one that discovered the CBD in 64 and the THC in 65. And then in the 90s, the anandamide, the endocannabinoid system, he's the god of this field. So Rafi is in Jerusalem, 1.5 hours from me. I called him. His son is professor in my faculty, in my institute, so I know him. I called Rafi and said, hey, I have this problem. I want to come alone. I came and he gave me all the bridle on one leg. He teach me for 4 hours. There are hundreds of cannabinoids, and there are also terpenoids, and there is flavonoids, and there more than 500 active compounds in the extract that I'm holding in my hand. So this is a huge problem. How you repeat the experiment? Does the plant that you get in the summer will be the same plant in the winter? The answer is no. I have a PhD in plant biology. If you change the temperature 4 degrees in tomato plant, you change more than 1,000 proteins that they express and chemicals and everything. So how can I repeat that and expand it? How can I go deeper? And with this problem, we started to develop analytical tools to do a better research. So first of all, we develop analytical tools to analyze all the phytocannabinoids. We can analyze today 144 phytocannabinoids from the plant. And then we went and we did the library for all the terpenoids. And then the flavonoids. Then we said, okay, now I want to know how it affects the body. So I need to look on the endocannabinoids. Today, if I'm taking blood samples, I can analyze almost 150 endocannabinoids from blood samples. And then we said, okay, the receptors. Everybody look on CB1 and CB2, but there are much more.
Sorry, can I stop you there just because I didn't understand?
Sure.
Did you say 150 endocannabinoids?
Yeah. Cannabis is a unique plant because it produces unique compounds called phytocannabinoids. But these molecules are unique because they are active on our body. And our body have the endocannabinoid system, which produce similar compound. And the endocannabinoid system in our body, it's a homeostatic system. It's a system that balancing many organs and many systems in our body. It's actually not an autonomic system. I'm not familiar with any cells in our body that don't have endocannabinoid systems. So almost all the cells in our body and almost all the systems in our body will contain the molecules and the active molecules of the endocannabinoids. And the endocannabinoid system, it's regulation and homeostatic. It's like a mild system in every system that bring back to balance. I can give 2 examples which I always give. One is how we feel appetite. If we are not eating for a whole day or for 2 days, our intestine is shrinking and send signal to our brain, send hormones, which will be very severe. We will be angry, we'll be hungry. We feel the hunger in our body. We can't concentrate. We just want to eat. And the reason it's very severe, after 2 days, our body will start to break muscles in order to produce energy. It's called kachektia. And for that reason, he's acting very severely. Think about the hunter 5,000 years ago that ran after the deer. After 2 days that he's not eating, he don't care about anything. He don't feel pain. He just ran and he need to hunt it. He concentrate on one thing. This is the severe action. But now there is come the mild action. The hunter succeed to hunt the deer. He put it on the fire. Now he's starting to eat. After few bites, he will feel full. But the body produce appetite that he will keep eating. You don't know where is the next meal. But now he's at full control. Think about the burn meat is also activate the smell in the, the pumas and the tigers and the wolf. And they
all
will
the people
come.
From the Lion King.
Yeah, they all have endocannabinoid system, right? It will activate all of them. They will come. But this hunter now is in the full control. He ate. He's still eating because he feels appetite. But if he will hear the noise, he will run away. This is evolution. The one that didn't run away just didn't survive, and he's not with us anymore. So this is a mild way to increase appetite. We can say that the same thing about pain. There is a severe action. If we cut our leg totally, if we wound harmly, our body will produce opioids like morphine, right? Which will reduce this pain dramatically. But there is a penalty. This is a severe action. We will be totally stoned. We will be out. We won't remember and we won't be in focus. But now, if we just hit our leg in the corner, it's just locally, we need to reduce this pain. This is the endocannabinoid system. So when we're touching a hot glass and our hand is jumping from that, it's because in our tip of our nerves, there are sensors for pain. This will send a signal to our brain to contract our muscles in our hand, and our hand will move from the danger. But from that point, there is no reason for the body to produce this pain. It will reduce this pain by releasing endocannabinoids locally in our tip of our fingers. And this reduces very fast the pain. But now it's locally without affecting all our body. But it's also very mild. If we will have a severe injury, it's the opioid system. If we have a small pain, it will be the endocannabinoid system. So I need 9 hours in order to go system by system and explain how the endocannabinoid system is active. The cannabis is just mimic that. It will bind the receptors and activate it. So the endocannabinoid system contains majorly 2 compounds. One, we call them endocannabinoids, which are small molecules, lipophilic one, that will release and bind another cell and send a signal. And the other cells need to read these signals. So we will have a receptor, which is a protein, that will read it. So these 2 major, we also have enzymes that regulate that, but I will skip that. So these major 2 compounds, endocannabinoids and receptors, usually people know about major 2 endocannabinoids, 2-AG and anandamide. But this is flat as same THC and CBD. There is more than 100 endocannabinoids. So today my lab have the ability, if I'm taking blood samples or I'm taking biopsy, to look on these endocannabinoids, we can analyze around 150 endocannabinoids. So 87 of them is 100% endocannabinoids. We have another 50, we call them endocannabinoid-like. So they look like endocannabinoids. I think they're endocannabinoids, but nobody published on them ever. So if I'm looking on the receptors, my lab is working on 28 endocannabinoid receptors. Usually people know CB1 and CB2, but it's very, very flat. We have other receptors that are participate in the endocannabinoid system, cannabis affecting them, and the endocannabinoids affecting them.
Now, one of the things that you mentioned earlier was particularly pertinent to a number of conversations I've had with other Israel-based researchers in the past, where it's been mentioned that consistently getting access to the exact same genetics and the exact same strains of cannabis to perform research on isn't always possible, and instead sometimes replaced with strains that are very similar in profile but not exactly identical. Now, with that in mind, what are the implications of this on studies that are going on in various research labs like your own, and how does that impact the findings that are taking place from the studies in labs across the country?
So I solved this problem to myself as having the ability to analyze all the active compounds, all the cannabinoid, terpenoid, flavonoids. I have a pattern. When I'm doing the experiment, I know exactly what are the compounds. And then I can repeat the experiments even if I need to combine strains or to change or to add. I can play with it. So this is the reason I put a lot of effort in order to do that. Also, when I have that, I can narrow down and find what are the accurate compounds that are doing the effect. And for finding them, then I'm not related on a specific strain anymore. It's a lot of work to be there. But from the point you're there, you're not related on specific strain. More than that, I can tell you, let's say that I'm finding a big finding in Israel. I see that the white widow, I won't say white widow, I would say more uniquely, a strain called Tchelet in Israel is very helpful in multiple sclerosis. How it's helped for a person in California, how it's helped for a person in Toronto. The same one, even if you take the same genetic, the Tchelet, and you grow it in California, you will get a different pattern of cannabinoids because it's a different soil, different sun, different temperature. Everything will be not the same. So even with the same genetic, you won't get the same pattern. So without the ability to tell you, hey, look on the strain that have these 5 cannabinoids in this ratio, we can't really collaborate. We can't really transfer knowledge. And this is an obstacle that we are crossing now when we have the ability to look on that level.
So you mentioned multiple sclerosis in your answer, and I wanted to move on from cancer and cannabinoids because I know that's where you started your research, but it's actually moved a lot broader. So, for instance, in the news recently, there's been some articles about cannabinoids being effective for MRSA as an antibiotic. Mm-hmm. I wonder what other areas of cannabinoid medicine are interesting you and your facility right now?
So we have research in my lab in few areas. We are very strong on neurodegenerative diseases like Alzheimer's, sleep disorders, multiple sclerosis, epilepsy. These are the 4 main topics. In all of them, we have mice model, cell model, and clinical trials running. We are participating today in 14 clinical trials. Another area that my lab is focusing on is how we affect the immune system versus its autoimmune disease or just immune system. We submitted today a paper showing that in immunotherapy, cannabis is actually interfere with the action. So what cannabis is doing in the immune system in general, very general, because when we're saying cannabis, there are strains that are doing and strains that are not doing, we should be careful about that. But in general, cannabis is tuning down the immune system. So when we have hyperactivity of the immune system, the cannabis is tuning it down. So again, bring to balance. If you're a normal person with a regular immune system that is not activated, that you just in normal, the cannabis won't affect it. But if now the immune system is in hyperactivity, let's say you have kind of inflammation or whatever, the role of the endocannabinoid system is to call the soldiers back to the office after they finish the battle. So what they are doing, they're tuning down. This is the role, the native role of this system. So let's say you have a scratch in your finger and there is a little bit, bacteria getting in. So now the body realizes that there is an invasion and he sends his soldier, he sends his immune system. So the macrophages, the NK killers, the T cells, they're all reaching the area and starting the fight, right? The one is going around, one is getting in, doing their job. And now they need to tell each other, hey, in my side it's clear and in my side it's clear. And they're announcing that by sending endocannabinoids. When everything is clear, they will leave the place and go back. Okay? This is the endocannabinoid system. So the cannabis is doing similar thing. He's sending this message to the immune system. So it's tuning down the immune system. So in autoimmune disease, when our body attacks himself, the cannabis is perfect because it tuned that down. But when we need the immune system, when we want to activate it, like in immunotherapy in cancer, you don't want to tune it down. So it's actually a conflict between the immunotherapy treatment and the cannabis. So my lab is working around that as well. I want to give one example from the cancer, how we are working and how we are looking on all other aspects. It's true for multiple sclerosis, it's true in the immune system. So we try to look to be very focused on one question and very specific, what we call in general, personal medicine. So we look on cancer, we not look on white cancer. We look on leukemia with a specific mutation in specific areas that activate and cause this leukemia. And what we're doing, we're starting screening a lot of different cannabis strains. In general, very basic, taking the flowers, doing extraction, and starting to do what we call high throughput screening. Started to screen. And we are looking for the unique things. We are looking for one strain that doing unique actions. So we look on this leukemia cells, screen 300 different types of cannabis, and just one of them killed the cells in very potent way. Said, okay, this is great. What is unique in this plant and why is killing the cells? We started to look on the cell biology to understand what's happening in the cells, and we realized that it's activating a pathway in the cell that is signaling through the ERs and the signal that not sending the protein to the right place. Never mind what, we understand the cell biology, why the mutated cells are dying and normal cells is not dying. After we understand that and prove to ourselves that this is a real action and we understand the mechanism, we're going to a mice model and we create these tumors, we're treating the mice, we see that we are reducing the tumors, we again understand the mechanism and everything. And if this is true, now we're asking ourselves, okay, what in the cannabis is doing this action? And we're starting to narrow it down. We're starting with, let's say, 150 compounds. We started to do fraction. We cut it to pieces. We said, okay, let's cut it to 5 fraction. If we have 150 compounds, 30 compounds in the first tube, 30 other compounds in the second tube, 30 other compounds in the third tube. And then we're taking this fraction, going back to the cells, going back to the mice, and look if one fraction is doing the action, or maybe you need 2 or 3. And in that way, we narrow it down until we find the minimum compound that's doing the effect. Sometimes it's 1 compound, sometimes it's 5, sometimes it's the whole extract, sometimes it's 13. But I, as a scientist, want to know what are the minimum compounds. So in these leukemia examples, we define 3 specific compounds, 3 cannabinoids, that you need the 3 of them, but you don't need more than them. If you have the 3 of them purified, they are working as the whole extract, killing these cells. And we already understand the mechanism. One is binding CD1 and activating the MAP kinase. The other one bind another receptor called TRPV1, release calcium from the mitochondria. We understand the system. But you need the 3 of them. Every combination of 1 or 2 of them won't do the action. You need the 3. One of these compounds is a unique compound, like nobody described it before. It's a unique phytocannabinoid that nobody published before. So what happened to us, and this is, kind of a proof of concept for me. 8 months ago, the regulation in Israel changed. We moved from, instead of the growers give it from their side, they need to give it through pharmacies, and they had to start to grow GMP level. So they had to change the greenhouse and to change the systems in the greenhouse. And after doing that, we're getting this strain, the same genetic, the same grower, and it's not producing these compounds anymore. So 2 of the 3 are there, but 1 is missing. And it's not working anymore. It's not killing the cells, not reducing the tumors. But knowing with which compound is it, I can go to the other strains in my lab. I have around 900. screen, doing bioinformatics, look which one is producing these specific compounds in high level, ask for the grower to bring it to me. We purified this compound, add it to the other extract, and we got back the same action. And again, this is kind of a proof of concept. We need to know in that depth in order to define and to repeat the experiment and to do all of it. And this is the way we are going with all other methods. It doesn't matter if it's in multiple sclerosis, it doesn't matter if it's in Alzheimer or epilepsy. And surprisingly, we are learning that we have a very complicated action with the whole extract. Not always it's synergism. So I will give you two examples. We all know that CBD is the major compound that reduces seizures in epilepsy. And there is even a medicine by GW, which is pure CBD, Epidiolex, that is a medicine to reduce seizures. So in our data, we see that there are strains that are working a little bit better than pure CBD. So if CBD will reduce 80% of the seizures, we have strains that will reduce 100% of the seizures. But we have one strain in Israel that have the same amount of CBD, the same ratio with THC, 20:1. We give the mice the same amount, 5 milligrams per kilogram, and it's increased the seizures. It's not reducing the seizures. So now the question, is it the CBD there is different because of some reason? Or maybe there are compounders that interfere with the CBD action. So if you will just look about the amount of CBD and THC, you will assume that this strain will reduce seizures, which is not true. So this is for epilepsy. I can give for the multiple sclerosis. We have strain that doesn't affect the disease at all. The same amount THC and CBD, exactly the same amount. Strain that doesn't affect the disease at all. We have strains that improve the disease dramatically, and we have strains that make the disease much worse dramatically. With the same amount of CBD and THC, exactly the same. So what we're learning from that we must look deeper. We must look on the phytocannabinoids and to learn the compound. We must learn how it affects the endocannabinoid system. Otherwise, we will keep doing trial and error.
Where do they go, the smoke rings I blow each night?
We hope you enjoyed this episode, and if you did, we have a slight request. Please do pass the pod on to anybody else that works in cannabis or is interested in the field that you think might get real value from listening to the podcast. And of course, if you haven't done already, please do like, share, subscribe, and leave us a lovely review. But for the time being, in the meantime, thank you so much for joining us again. It's been great having you with us on Professionally Cannabis, and until the next episode, Shalom.
Hello, please take me above, take me with you.